What I'm going to try and do for the next hour, 50 minutes or so, is give you kind of a look of where I've come from, some of my interests. Some of you know me, but most of you haven't a clue who I am, why I'm up to. Other than I'm very interested in genomics. I'm very passionate about genomics. And the other half of the talk is going to be more forward-looking, more aspirational, actually. I want to tell you some of the thoughts I have about UQ Genomics. Spoken to lots of people over the last, intensely over the last 6 months, but certainly since Christmas last year. So what I'm going to talk about, it's not just what I'm thinking of, it's what everyone else is thinking of as well. And as I go through the talk, every so often there'll be some slides that Ben Hayes, Robert Henry, and Joseph Powell and all the rest have given me. This will sort of illustrate some of the genomics that's going on around the university. Unfortunately, the pointer thingy is not working, which is good in one respect. It'll stop me wandering up and down. But here we go. So, I've got this slide here. Actually, a lot of my interest in genomics actually started in Australia, funny enough. Quite a long time ago. This was a meeting, I think, was it 1995, organised by Jay Hetzel, Steve O'Brien and Jennifer Graves. Very nice place, one of my favourite conferences. This was at Fraser Island, and it was, good. Excellent. It was the very first time I came to Australia. It was a great experience. It was a 3-day workshop organised by the Human Genome Organisation at the time. And there was nobody there working on human genomes at all. It was all about comparative genomics. In fact, it was about pre-genomics. This was all a time when we made these things. These are comparative maps. We would make a linkage map or some sort of physical map of, in this case, chicken, and then compare that with maps in human. None of us at the time thought we would ever get whole genome sequences, but a few of us kind of half wished it's definitely going to happen one day. But this kind of work was, the human genome was being sequenced at the time. In my lab, which was working on chickens, we were continually flipping back and forth between a human genome sequence as it was coming out and the maps that we were making in the chicken and start to compare them. We were able to, in this case, for example, predict the sex-determining gene in the chicken. And nicely, Craig Smith, who's at Monash now, and Andrew Sinclair from Melbourne, showed directly that what we predicted just by comparative mapping was actually true. So that was quite satisfying. The other thing that happened at the time, a lot of people said I was mad trying to compare a chicken genome and how the genes were organised with human, because it'll be completely different, completely smashed up. But actually what we showed was just with a few hundred gene markers, we were able to show very crudely at the time that the chicken genome was actually very similar grossly as organised as a human genome. In fact, it was just as good as human versus mouse. Until you actually got down to the nitty-gritty, the very details, then there were a lot of rearrangements. And this led on very quickly, obviously, to the human genome sequence. And many of the people at that workshop I mentioned in the very beginning were involved in this part, which is the comparative genomics side of the Human Genome Project. And the aim there was to be able to take sequences from diverse species and then compare them to human genomes and say, well, what's conserved in all vertebrates, for example. And many of those things were actually obviously genes and exons, but there are also things like that turn out to be enhancers and regulatory sequences that control the developmental, control the gene expression. So that's quite interesting. But of course, the rest of us were a bit devious. We were working in chickens and pigs and cattle and things like that. We were sort of interested in the human genome thing, but we're really interested in getting a genome sequence of our respective species. And that did happen. And it cost a huge amount of money and really labor intensive. This was kind of the way it was done in those days was shotgun sequencing, very, very, very labor-intensive Sanger sequencing. And Wes Warren, great friend of mine, has persevered all these years, and he's sequenced the chicken and continues sequencing it, as well as several other species. And of course, great papers, loads of papers came out, and this diagram here very crudely kind of shows you a comparison of chicken and human, and it just confirms, but obviously in great detail, what we predicted in that earlier paper about 5-6 years beforehand. There was a lot of similarities. And this was a period when we were sharing lots of data. We were very open about data. So this is the Ensembl genome browser. And I've worked with Ensembl for, nearly 15 years or so. And we continue to work with them. And now they're a great place where you can view this kind of data and you can share your data. But it's actually getting a bit out of hand now, as I'll come to in a second. Very soon after this early work, other birds were sequenced. First, the zebrafinch as a model of neurobiology, and of course turkeys were very, very popular in America, of course, and the duck, mainly from the influenza kind of outbreaks that we have at the time. But of course, we've all seen this picture before. This is sort of an update. Sequencing costs coming down, 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 down. And of course, like other people, I always like to add, it might only cost a few hundred. Well, I can sequence a chicken, a de novo assembly for about $300, from complete scratch. But it costs me quite a lot of money to employ somebody to actually put it all together and do the work afterwards. But the cost has come down in terms of numbers. And this kind of equipment came out a few years ago. This is the X10. We bought this in Edinburgh. And like I say, we can use this now to sequence chickens for a few £100. Yeah. And at this time, of course, loads of people got together, were interested, of course, in birds, particularly Duke, BGI, and various other places, and we all ganged together to sequence a lot more birds. So now we've gone up to about 50 bird genomes. And this was really nice. It's a nice example of groups with a shared interest in bird evolution, but also within there's lots of other interests about genome organization, about biology of birds, all kinds of things. And this group actually is continued, so currently there's nearly 300 genomes that have been sequenced. But I should add that here's a nice review by Alex Su Lab, where there's a phylogeny of birds based on that data. I think it's about 75 birds in this list. And you see these various columns here. Basically what it's saying is only one bird in there that is assembled at chromosome level assembly. That's a chicken. The zebrafinch and the hummingbird's kind of close second, but most of the others are all draft sequences. And Eric Jarvis did this little summary here of 28 of the birds originally. This is just done about a year or so ago. Basically showed that the scaffolds, that's the contiguous pieces of sequence put together into as long a piece of genome you could put together, were still quite small. And this is a problem of Illumina technology, short read sequencing. But many of you have heard, where are we? It's gone now. Yeah. About PacBio sequel system, which gives you a lot. The reads are much longer, 10, 15. If you size select, you can get up to 30 KB sequences. And over the particularly last 2 years, this has revolutionized assembly of genomes. Costs more though, it's the only problem. And this is Gull 5. It's the latest public release of the chicken genome. Again, it's done by Wes and several others, putting all the data together. Key thing here, some numbers down here. The N50 for the contig is nearly 3 megabases. and the N50 for the scaffolds is more like 7 megabases. So it's huge, long lengths of sequence. So this is quite easy now. So this is done with quail, hummingbirds, zebrafinch, several other birds. But like I say, it costs a lot more, at least 10 times more than the Illumina. But if you want a full length chromosome level, almost chromosome level, then you do it this way. But Wes has gone beyond that to Goal 6, which is not public yet, but basically it uses PacBio and actually combines with some Illumina reads. And this is a summary of some of the statistics. But I want to really show this here. If you use these High-C methods, this one's here from Phase Genomics, but there's also Dovetail, you can get complete chromosome level assembly. So he reckons he has every chromosome assigned with a complete scaffold, with not complete sequence, but at least a fragment, a framework of sequences along every chromosome, even the tiny, tiny microchromosomes, which are very, very difficult to sequence. So combining these various technologies, PacBio, Illumina, and the High-C methods, you can sequence through repetitive regions, regions with duplications, lots of repeats, that kind of thing. And it's just acknowledged people at this stage. Now, one thing I would like to do in Queensland is to actually bring all that technology here. With Robert Henry, we've applied for funding to actually do that. So who knows, in the next few weeks, we might hear that we have that. So I think we've got part of it. Essentially, that's going to be PacBio for long reads, optical mapping for physical reads, and then we can outsource the High-C methods to get the chromosome level side of things. The other side of a lot of work that my lab's done for many, many years has been gene annotation. What I mean by that is, where are the genes? Where's the transcripts? Where's the coding genes, non-coding genes? Where are all the regulatory sequences? This is sort of common to what we're all interested in all the different species. So years ago, this is a standard Ensembl pipeline. So like I said, we worked with Ensembl for many years. So we basically hand them data, and then they try to model genes to present them on the genome browsers. So about 5 years ago, they updated their homology routine, which is mainly based on proteins, to include short-read RNA-seq data. I think the thing's going to die on me. Short-read RNA-seq data. And this was great. You could actually get experimental evidence for the expression of each exon. So you could get a nice intron-exon structure of your gene, but it's very difficult, and I would say impossible, unless it's a really simple gene, to actually determine the structure of all the different transcripts. And that's really a limitation of the short-read methods. This is, at the time, this was a few years ago, this is a comparison of human and chicken. And you see chicken had about 17,000 genes that were predicted, and about a very similar number of transcripts. And of course, we all know that's rubbish. We know there should be a lot more transcripts. Human, a lot more manual efforts gone into the human genome. See, nearly 200,000 transcripts and about 20-odd thousand genes. That sounds more sensible. So something's missing in the chicken, and in cattle, and in pigs, and goats, and all the other species. So this is just a quick comparison between chicken, the kind of the types of RNAs you get, and in human. It's obvious, lots of stuff's missing, and it's mostly the non-coding part, non-coding RNAs, I should say. So a couple of years ago, we worked with PacBio, developed an ISO-SEQ method, mainly some of the simple methods in the computational procedures. And this diagram here just illustrates the problem. With short reads, you've got to try and piece them all together, and it's well nigh impossible to get an unambiguous assembly of a transcript. But with the ISO-SEQ method, It's very simple. All we need to do is clone a full-length cDNA with a 5' cap and a 3' poly A, sequence that completely. So we don't need to assemble it, we just get a full-length transcript. So my analogy is a bit like Richen did 15, 20 years ago, just made full-length cDNA libraries. So completely independent of the genome, and so now you have this collection of transcripts you can now just simply map onto your genome. Nice thing about this is, because as you all know, genomes are in flux. Hopefully, the next couple of years they'll be finished, but they're always being updated, and it's a real pain having to take your short-read RNA-seq data and map it back and assemble it and redo everything. But this procedure here, we have a permanent resource of full-length cDNAs, and you just take them and map them onto your genome every time a new assembly comes out. So this is just quickly a quick summary of what you get. These are the results that we found. So again, at the time, that was version 85, it's a couple of years back. Let's just reiterate what I mentioned earlier. About 17,000 genes, not many more transcripts. This is just looking at a brain and an embryo. We find, for that particular two tissues, we get 14,000 genes, coding genes, 17,000 link RNAs or long non-coding RNAs. You notice up there aren't any in that particular version of Chicken, because nobody could get a reliable assembly of any of the long non-coding RNAs. So that was quite an advance. 64,000 transcripts. So now we're getting a bit nearer the real world. This is the latest version of Ensembl. Many more genes, many more transcripts, and the reason for that is they've used all our data. They're still too conservative. They collapse a lot of their transcripts, so we have a sort of a slight disagreement on how to actually annotate things properly. But show you a pitch, what you got now on there. Those red bars indicate frequency of transcripts, genes, and the number of exons. So now you see a lot more multi-exonic genes. This is the best picture. This gives you a really nice snapshot of the type of thing that you see. This is just for one gene. The very top, you'll see an intron-exon structure of a gene. That's the traditional Ensembl view. One transcript. Using cufflinks to assemble some RNA-seq data, you get that dark red line underneath. A little bit better, you get an extra exon. But when you use the full PacBio sequencing, you actually get a much better picture. You get this really complex picture of transcription, multiple starts of transcription, and we know these are all different start sites because they're capped. And all sorts of alternative transcripts. And at the end here is quite interesting. Both the cufflinks methods fuse this transcript to this transcript. That's actually an error there. This is actually an independent transcript with a transcription start site here. So this is a long non-coding RNA at the end of this particular gene. Of course, now, what do these things do? Who knows? That's the next question. At least we got a more accurate picture of what's going on. So to get some idea of how many transcripts, how many genes there are, we actually did a standard cufflinks analysis using 20 different tissues from short-read RNA sequencing. And we predicted 50-odd thousand genes and over 200,000 transcripts in the chicken. And last week, my lab in Edinburgh, we still work with, just finished analysing 6 different tissues, normalised libraries, full-length cDNAs, and we have 52,000 genes in the chicken and 471,000 transcripts so far. So it's very, very complex. Chicken's just as complex as any other vertebrate. So we have a very accurate picture now, and so now we can use that and go on and start asking questions. What do these things do? So I'm going to acknowledge all those people there. That's my lab in Edinburgh. And PacBio has been very, very helpful for years in this project. The other area is comparative genomics. Like I said, it takes right back to my interest in the very beginning. Very simple idea. Let's take our sequences of different species, line them up in a multiple genome alignment, find blocks of conservation, and start to say, what are they? Well, they might be exons, but then they may not be exons, they may be regulatory sequences, that kind of thing. So this is work of Leroy in my lab. You basically use the phylogeny of the 50 bird genomes and all the sequences. And you can do two things with it. You can discover proteins, and you can discover non-coding, non-transcribed regions. So the first coding part, this is easy. You all know the genetic code is 3 base pair code, and the third base is usually a wobble, and the first two are reasonably conserved. So in principle, you can imagine you can go through a genome sequence and look at conservation, and you can actually tease out where the coding regions are simply on looking for a triplet kind of periodicity. Whoops. And this is based on work with Erwin from the Broad Institute, based on his phylo-CSF package programs. So here's a nice example of a typical result. So you see that this part here is the coding region, and then either the 5' or 3' end of it has lots of stop codons and all sorts of things like that, and it's not very well conserved, etc. So that picks out very nicely an exon and a coded sequence there quite easily. It's a bit more interesting here. What you see here is you see an open reading frame, but it doesn't stop at the classic stop sites. It actually goes through about 3 different stop codons, and then finally stops here. And this is all kind of non-conserved. So that's what you call stop codon read-through. So actually, when you look at the coding potential of a genome, it's not as easy, as simple as you think. It's not simply an ATG and a stop codon. Life's more complicated. You get different starts, translation start sites, and different 3' stop codons. So you can get different lengths of proteins. Using this procedure, you can actually see not only a single open reading frame, you can get overlapping open reading frames. You can detect small open reading frames. So when you look at those long non-coding RNAs, you can see some of them actually a code for a very short peptide. And we've kind of dismissed those before. So I haven't got time to go through it, but we see all those features in the transcripts of the chicken. So many of us ask this question, oh, okay, it's easy to find transcripts. It's easy to find coding regions. What about the rest of it? What about the regulatory areas? So if I have a genetic mutation, a deletion, or a SNP, that's not in those obvious parts, which is only a few percent of the genome, how can I actually predict that my deletion affects, say, a promoter region or whatever? Well, looking at conservation is one way of looking at this. This is just using those multiple alignments and looking for, basically all you need to know that these orange bits that come up are regions that are in selective constraint. They're evolutionarily conserved. So you can actually go through and look at these, and you can identify blocks. Some quite short. If you have enough sequence, you can get right down to a single base pair. But in this case, we're down to about 10 base pairs, and you can go up to, obviously, to several kb, depending on how well conserved it is. You can detect every exon very easily, and that's just shown with those red blobs. Those are ensemble predictions, and they overlap quite nicely. But there's these black constrained blocks. But not all those black constraint blocks overlap with a transcript, so they're quite interesting. So Lel's used this procedure, and just look at this one here. This is a 49-way alignment using a program called GRP, and you can find nearly 1 million of these elements. Nearly 20% of the genome is under selective constraint. So if you're going to say, where should I look in the genome for something interesting, look there first. But I'll come back to that. It's not, everything's not in there, but a lot of it is in that 20%. So just to illustrate, using this, at the top there is a bit of a chicken genome, and the black boxes indicate constrained blocks. So what are they? So next one, we overlap with some transcripts. These are the classic ensemble transcripts models. So you can see, really nice how these blocks overlap with, in this case, these red parts of the coding exons. And you see the same here, really nice. An extra exon in there maybe, or regulatory sequence, I have no idea. Lots of blocks here, no annotations at all. So now we overlay on top of this all the transcript data that I mentioned earlier. So now we find, again, a lot of these blocks come quite nicely over here. You notice there's nothing transcribed for that block, so that could be a regulatory sequence. This area in the center here that had no annotations at all actually is another gene. So that, and you see all those blocks, they overlap exactly with all the exons. So you can discover new genes. Here's some more data. Up there is these histograms plot the transcription start sites as mapped by CAGE-SEQ, which just quantifies transcription initiation. That quite nicely shows us the transcript here, overlaps to this bit, and this new transcript has got an initiation site here, and this one here has an initiation site, and there's also some weak sites down here. So you can see what's happening now. We can overlay all kinds of data on top of our genome and start to annotate it and figure out where all the genes are, and also regions that might be involved in some other kind of function. So Lel Eri is the person that's done most of that work. So last of all is gene regulation. And you all heard of ENCODE. It's a huge project in human genome, trying to map out promoter sites, enhancers, lots of epigenetic binding factors and things like that. Well, in my case, Steve and Ben, we're interested in farm animals. And there's a similar project. It's more of an informal collaboration between labs all over the world, trying to use these kind of methods here. So I've talked about RNA-seq and CAGE, but also all these sort of chip-seq methods, attack-seq, etc., to define potential regions involved in gene regulation. And this is under the FANG consortium. Here's just a snapshot of something we've done in chicken. Again, it's this complex sort of series of tracks. So Lel's developed that with Ensemble track hub system, where you basically put all your data in, and then you can overlay it on transcripts and genes, etc., etc. So here you see the black blocks. Again, are those conserved blocks. Here's a transcript. Here's a set of chip marks. And up here are CAGE sequences. And this is basically a bidirectional promoter that's active in this particular tissue. But it's going to get quite complicated because you're going to get more levels of data. But using hidden Markov models, you can kind of go along and group these various signals together and say, that's a promoter, start site. That's an enhancer. It's all predictive. but then you have to do experiments to prove it. So Leo's done that, gone through all the data, and it's limited because he's only done a few tissues. But here's a histogram of, for example, here's exons, and you can see that there's a frequency of exons, and in light blue, about half of them are in constrained blocks. So what does that say? Well, the other half, we don't have enough data yet to actually say they're constrained, because all the regions are in the multiple alignments may not be equally covered, or these are exons that are rapidly evolving. And it's even more obvious when you look at promoters in enhancer regions. There's plenty of them, and they're very rapidly turning over probably, so they're less conserved. But at least we've got a start to actually identify some of these things by looking at conservation. So this brings me to the other half of the talk. So it's all about genome information. So everything I've spoken about so far is about genome assemblies, annotating those genomes. I haven't talked about it, but also looking at things like intermediate phenotypes, where we're looking at expression atlases, transcripts. But it's very, very important that we go beyond prediction and we look at functional genomics. We actually test. I think this element is doing X, and if I mutate it, I can then start asking questions about that. Because we're all here, really, whether we work on chickens, fish, wheat, sorghum, bacteria, it doesn't really matter. We're all here doing this kind of work, and we want to end up understanding how this sequence and its variants on that sequence can be translated eventually into the phenotype of the whole organism, or what those various proteins and genes and transcripts are doing. So that's why a couple of years ago, there was a lot of debate in University of Queensland, and the UQ Genomics Initiative was put together because these are the things I've talked about are common to all the things that we're interested in, not just in one specific species of medicine or in crops. We use those same methods, same kind of resources, but we just apply it to different problems. And that's really the gist behind the Genomic Initiative, is to try and draw these things together, more coherent view and less fragmented. So the UQ Genomic Initiative is really a one-stop shop. It's what we want to see. So for example, the UQ Genomic community now has a voice. So rather than being to like in 20 different places, you can all kind of gang together. This is my hope anyway. And what do you need to do your research? Let's talk about it. If government, or if the, well, let's work our way up. If the university, or if the local government, or the national government want to speak to the UQ Genomic community, then they know where to come. They can talk to this group, rather than having to talk to lots of other places. So that's the kind of general idea behind the initiative. And the university wants to obviously invest in this area and have a high impact on many areas. I've really spoken so far mainly about the genome information, the technologies, that type of thing. But the other three areas are in the obvious major areas: health, food, and the environment. So I'll give you some examples of sorts of things going on around the place. So here you can kind of look at this in different layers. I think most important are the people, skills, training, that type of thing. And then you'll use a set of facilities, generate a huge amount of data, you analyze it, that information is presented, and then we need to exploit it in different areas. And the three I've talked about. I've also put up the Genomic in Society because there's as you'll see, there's huge implications, legal, social, ethical, in this kind of work. So, people. I personally believe that the university needs to invest in people. Training is very important, and it doesn't stop when you leave university or college. It goes on until you retire. So I want the university to actually support training, for example, going to informatics courses and things like that, all the way through, and not make it something that's difficult to get into. This is just a very brief list of the kinds of skills, interests around UQ. Medicine, ecology, crops, biological sciences, genome sciences, and the ethical side of things. Huge amount. This is just another list of institutes. So a large number of institutes all over the place, all disconnected. So we clearly have a lot of skills, a lot of expertise, a lot of resources in the university that we need to kind of draw them together. At least if you know they exist, that will be useful. Facilities. Created a facilities group, and you'll see a number of groups that we're trying to put together. Again, I want you, as being members of the genomics community, to have power to say what you want, as opposed to coming from top to bottom. I'm a bottom upwards kind of person. So for example, the facilities group, its aim is simply to ask things like, what do we need to do our research? What do we have at the moment that's not working? How can we improve it? If there's an opportunity to obtain funding for some equipment, what do we want to buy? What do we want to get? So for example, 6 months ago, Robert, Henry and I and a few others put together a LEAF proposal. Next year, when that LEAF proposal comes along, I would like to see virtually on the shelf that the UQ Genomics community actually have already got a list of things they want. Right? So you don't have to wait. You can respond to this very quickly. One of the things that's happened over the last few months is Steve Love and a few others have tried to bring together some of the facilities that we already have. Some of the equipment has been in boxes, in cupboards, and just sitting there doing nothing. So some of it has been brought together, and the UQ AGRF Integrated Genomic Facility has been created to provide access to everybody for some of these facilities, sequencing, etc. That will grow as we get more equipment. I talked about sequels that will be located there. But it's a UQ stroke AGRF facility. That means is that we, the university, have invested in that setup. So you, as a user, should get preferential rates. So you will. And the person that's key, well, obviously Ken runs the facility. Jeanette is really good at those very difficult procedures. And we need to invest in people like that to do all those sort of technically difficult libraries I talked about earlier. But Brooke Gardner has a lot of experience working with all the difficult researchers in the university over the last 15 years or so. She knows what's going on around, and she's going to be your genome coordinator. So she's there not as an employee of AGRF, but she's there as your representative for what you want. I'll come to it at the end of my talk, but next week we'll have a 2-day meeting to launch the Genomic Initiative, and Brooke will stand up half an hour or so and tell you a bit more about what the ideas behind this facility. But it's not just the IGF. We also have other facilities. We have the ACE, that's the Australian Centre for UQ Genomics, and IMB have a lot of facilities as well. We don't want to destroy those. We don't want to merge those with anything else. They will continue. But what I want to see is that they all work together. So, for example, trivial but very important, instead of having separate accounts with Illumina, we have one. And then you get everything a lot cheaper. So you get cheaper rates and discounts, things like that. So more of a virtual core facilities at the university that all talk to each other. Okay. I've got a couple of slides here from Joseph Powell, which illustrates again future. But actually, he's doing it now. One of the major areas in genomics today is single-cell genomics. To actually look at single cells, the genome, the transcriptome, accessibility of the chromatin, all part of other approaches. And it's very technically difficult. Very, very difficult. Joseph's got the equipment there. He's got lots and lots of collaborations, but that's not sustainable forever. It's quite limited to how much you can do. So I personally want the university to invest in this in terms of staff and supporting it, in which case we can then open it up to other groups that want to use the same procedure. Walking around the university, I talk to people, and I hear lots of examples of people working on or wish to work on single cells, and they don't know each other. So again, this is a way of bringing it together. And Joseph's working on this, which I think is absolutely amazing setup. This brings together single cell genomics and genome editing. Principle, it kind of sounds easy. What you do is you express a guide RNA in a single cell, it knocks out that gene, and then you have subsequently an effect on gene expression of that cell. So you then assay that. But of course you get a whole library of guide RNAs into this system, and so you can knock out different genes in single cells, and then you can start grouping them together. So it's like a whole mutagenesis procedure. So it's absolutely incredible. So this is called different names for it: crop-seq, perturb-seq. So over the next year, this will be operational, I hope. Again, it's huge potential, but to get the most of that potential, we need to invest in it. Informatics, I mentioned this before. All these procedures produce vast amounts of data. Again, we want to work together on this, so created an informatics group. I call it informatics rather than bioinformatics on purpose. Informatics to me is the whole suite of things. It's bioinformatics, it's statistics, it's big data integration, it's all those 3 Bs, okay? Kind of work we're talking about, we all do this kind of stuff. From the very beginning, if you take a genome, you sequence it, you want to find variants in it, that could use established procedures. So we need a forum to discuss several things. One is the hardware that's available around the university. Is it up to what we need? Sometimes it is, depends what scale you're doing. Other times somebody comes along and says, I need this vast amount of memory and processors, and it'd be great if you could actually forecast that, because then you can then work, say I want that next year maybe, because we all know we want more and more. So we can kind of, through this group, we have a voice to actually predict. So when David Abraham comes along and says, what do you need? I can say, off the shelf, this is what we need, okay? Not, oh, well, give us another few weeks, we'll tell you what we need, okay? We also need access to, bioinformatics is a lot of workflows. It's actually putting together lots of established methods into a workable pipeline. In many cases, that's fairly robust. Some people are quite good at doing these things, but they can just be handed around to everyone else. So you should have access to that kind of thing. Skills, training, big deal. It's number one thing that was mentioned to me, virtually every person I spoke to over the last year was training in this area. So it's hardware. Also, This group can have a role in what's relevant to Queensland. So if, for example, if we get some requests from the Queensland government about what do you think of cloud computing or something, okay, we were the group here that can actually talk to them and say this is what we think about it. Similarly, on the national scale, I mean, Dominic Gorse and others are involved in developing ideas about a national program, a national roadmap for bioinformatics. So again, you have a role in that. So like I keep saying, get involved. So I mentioned skills and training. Dominic's involved in a lot of this. And Nick Hamilton, you keep seeing all about hacky hour and things like that. There's some fantastic things that go on site. I'm still amazed that some people don't know these things happen. They should be available for everybody. I like this, the idea of a virtual lab. Some labs do massive amounts of bioinformatics. They have staff and trained and all the rest of it. They're well equipped. Actually, they can get on with it anyway. But other labs might only want to use these procedures for a few months of the year. So they need some way to be able to get access to these tools. Many of the tools are fairly routine, but they're kind of tricky often to actually to use. Mainly because they're all command line and things like that. And Dominic's team have put together these virtual labs to try and simplify that. And one of the mechanisms is through the Galaxy setup. So again, it's the thing that we need to invest in. Again, it's the thing that what do you need? Is it something routine? Can we adapt it? Can we put it into this virtual system to get it to work? Because sometimes it's simply a plumbing exercise, bring things together that work in a sort of pipeline with a nice front end. Okay. I personally believe that I'm not being derogatory here, I don't think some of the bioinformaticians or computer scientists who are in should be wasting their time on some of this routine work. I'm not insulting anybody, but using a virtual lab system, it means they don't have to. It means they can concentrate on things like developing new algorithms, developing those pipelines and testing out pipelines and things like that. So their time is better spent in that area. I think that every geneticist, from undergraduates, postgraduates and beyond, should be able to do this type of work. My old lab, about 10 years ago, I switched to everybody in the lab, apart from a couple of technicians, could perform, were proficient on Linux machines, and they could perform all this command line kind of work routinely. And that should be what we should be aiming for. So let's get at the university now. You should have courses that everyone's taught on this, and it should be mandatory in genetics. This is quite nice. This is something put together by Jeff Christensen. This is as part of plans for this roadmap I was talking about. Don't need to look at the details. The main idea is trying to collect together the sort of information, the sort of tools that you'll want to use. I mean, it's the things like, here we sort of create data, and then we analyze data, and then there's that big problem of trying to join data together, the big data integration problem. And then we submit data somewhere, and then we publish it somewhere, okay? These are some ideas of how to do that. So again, I think Dominic has circulated a survey recently asking you, what data sources do you want to get your hands on? What tools do you want to get your hands on? Again, it's what do you want? Don't struggle. Tell everybody what this group, informatics group, what you need, and let's get it. You'll get used to this. I am not a person that says you can't do something. I'm a person that says you can do it. I have no idea how to do it. Let's figure out how to do it. So it's a different way of doing things. I think over the years I've come to the conclusion that anything's possible. I say to my PhD students, if you're lucky, it's going to happen during your PhD. And I had a student years ago Her PhD was the aim was to sequence 10 different species of birds to get some innate immune genes. She started her PhD within a month. We had 50 genomes. She didn't need to do that. And she switched to, I sent her on courses for 6 months to learn how to analyze this kind of data. So she was lucky. She was at the right time. So we've got agriculture. This is a picture from Robert Henry's slides. Huge amount, billions and billions of dollars of income is generated in Queensland alone in crops and livestock. Plant genome sequencing, I'm going to run out of time here. Plant genome sequencing around the world. This is just a diagram of a couple hundred genomes that have been sequenced. Most of those are actually related to crops, various products. Of course, this is a plant phylogeny, and it's difficult to see, but basically we need to get more genome sequence around that phylogeny to be a bit more representative of plants. And there are a number of international projects going on. Now I would say, again, through the UQ Genomics community, we should be connecting up with not just national consortia, but international consortia all over the place. I don't want to duplicate anything. It's best not to. It's best to join up with other people, and you get the best out of things that way. So there's projects here looking at 1,000 plants. Why not? I like this this thing I grabbed on the internet. Great picture of Robert with his hat on. I think he's in Northern Territories, maybe. I like this story. It's about, first of all, the scientific part. It's about discovering something about the history of domestication of rice. And you would have thought that in northern Australia, you would actually get crops, plant species, rice species, that told you a bit about rice. And I always sort of traditionally think of that as being in Asia. But the great thing about the story is the only time you can study these things is when it's wet, when there's crocodiles everywhere. So I think that picture there is probably surrounded by crocodiles in the Northern Territories. Tricky work. This is quite nice. This is some work from Ben Hayes. Again, it illustrates cattle genomes with sequence. In fact, recently, last week, a new genome came out. Lots of markers, so you can quite simply do gene associations now in cattle. And of course, in Queensland, you have millions of, well, hundreds and thousands of cattle on various ranches. So the phenotypes are there. It's just the technologies around to do the sequencing. And Ben with Mike Goddard were instrumental several years ago in developing these ideas of genome-wide prediction, which now are used all over the world in not just cattle, chickens, pigs, all the commercial breeding, and probably plants as well. The idea, principle, simple, complex to actually to get on with, but you have a reference population with your phenotypes and your genotypes, and you create equations to predict phenotypes from a genotype. Then you apply those equations to your target population, where you only have genotypes now, and you can. The idea is you want to accurately predict the phenotypes of those offspring. So it's particularly useful for things that are high value, hard to measure, expensive traits. But Ben has gone one step beyond this, brought this in just to highlight microbiomes. So this is another area of expertise at UQ. Over the last few years, you've heard a lot about your gut microbiome, for example, influencing your phenotype, your immunity, your digestion, that kind of thing. So he went ahead and tried to test this in cattle. And the bottom line is when you you combine the microbiome information with the genome information, you get reasonably accuracy. You increase in accuracy on predicting, in this case, I think it's feed efficiency. So it's a nice example of bringing these two areas together. I definitely run out. Ecology, lots of areas here. Again, there's the ACE facility, but I kind of highlighted some here that Mark Reagan gave to me and Peter Mumby. It's about, obviously, one of the big things about Queensland is the Great Barrier Reef and how it's being bleached. Can genomics actually help there? Well, one thing at labs in Queensland now, so now this is more large-scale collaborations with, certainly with James Cook University and various other places, to look at those communities on the reef, on the reef, and actually sequence them to find out what do they consist of. So again, it's a complex interaction of corals and algae particularly. So we believe, again, if you kind of think of that work from Ben Hayes, it should be possible to sequence these algae, for example, look at genetic variation in those algae, look for algae that are tolerant to heat, for example, certain parts of north to south on the Great Barrier Reef. Use that information then to select by genome-wide selection on algae that are going to be adapted to higher temperatures. So I definitely think we can use those principles in agriculture and genomics to solve a problem in ecology, and certainly a major problem in the Great Barrier Reef. And those sort of ideas, hopefully, are going to come off when they get funded. Last of all, there's health. Many of you have heard of a lot of these genome alliances. Well, this is just a diagram I borrowed about the Australian Genome Alliance, which started a few years ago, Melbourne Genome Alliance, Sydney, and the last one was the Queensland Genome Health Alliance. And the idea here is to take all that research in the medical research labs and translate it and implement it in hospitals. Again, this is another one that's not unique to Australia, so why reinvent the wheel? So these groups are working together with the GA4GH, Global Alliance for Genomics and Health. There's about 60 odd countries involved in this. And they're looking at different aspects. Of course, there's the clinical, the data management, the ethics, and of course the security. Again, that brings in all this ethical ideas. Again, it's not new to one, it's not unique to one place, so we should be involved in all these things. David Bunker is the Executive Director of the Queensland Genomics Health Initiative. $25 million. Basically two parts, demonstration projects and a set of capabilities. Just giving one example, there's Peter Sawyers looking at melanomas. Of course, Queensland is the hot spot in the world for melanomas. So he's trying to come up with a way to stratify people that are high risk of genetic causes of melanomas, susceptible melanomas. And then to, once you stratify those, to actually look at those and screen those people to try and avoid them coming down with melanomas. So it's quite interesting. I like this slide. This is one given to me by Grant Montgomery. Again, it's sort of going right back to that slide about genome to function. This is what this is about. So we do a lot of this work here where we identify genomic regions that look interesting, some way or other, maybe functionally or maybe by genetics, and we look at genetic variants, and we can do things like find which genes are involved, but usually that's where things stop. Okay, so we need to move on and look at functional studies, and this is a medical slide, of course, and clinical outcomes. But it's common to agriculture, it's common to everything. And the UQ3B, or the UQ Project 3 billion IMB, is involved in this kind of work. A whole bunch of people with different expertise. What they're trying to do is solve this problem, sequence to function. Basically using models in genome editing. So at IMB we've got Peter Koopman making and Neil Nathans engineering mice and cells. We've got Ben Hogan engineering zebrafish, and we've got Ernst Wolftag creating iPS cells and organoids. Well, all of this kind of work can happen in other species, not just humans. So let's open it up. So again, I would like these things to be supported by the university. There's this Australian Functional Genomics Initiative. Again, those groups should be involved in that. And they're only going to be involved in that if we have the support to do it. So that brings us finally to the Genomics in Society. Karen Hussey has a group that's supported by a couple of sources, 3 sources, to look at this, look at the ethical, legal, societal impact of genomics. And I just highlight this article came out last week. It was a bit disturbing. In Australia, if you have a genetic test, maybe you've had it because you've done 23andMe, or maybe you've been part of a research project or something like that, and you're shown to have a defect of some sort, then you may not be able to get insurance. I think that's really bad, because we want a system where we want to prevent preventative medicine rather than treating the causes at the end. So again, that's something that could be discussed in great detail. We need a genomics communication group. It's obvious to me in the last 6 months that a lot of people don't know what's going on, even just across the road. So we need to improve that by websites and the like. And last of all, we need a genomics forum. And next week we will all get together, I hope. And genomics forum really is a place, it's a grouping, it's a way to get the genomics community to speak to each other. And next week, I'm really pleased that Marie Mangus has helped me to organise a 2-day symposium. We've had volunteers from all over the university to give talks, so I hope many of you will attend this, but it'll give you a better idea and a way to meet, and we're also going to get to meet people and network, that kind of thing. And I would like this to happen frequently, but it all depends on the genomics community, genomics forum. How often do you want to meet, right? So maybe once a month, once every 2 months, who knows? So last of all, just a couple, some of you know that I have an interest in combining science with art through photography. So this is where I've come from. This is Scotland. Those cute sort of puffins, sundews. And it's great to see these kind of things locally. So these are some pictures of some of local inhabitants I've picked up, photographed just around here. Great, these spiders. And lorikeets, absolutely gorgeous. And this, and the lizards, brilliant. Walk right up to them. Yeah. So I know I've gone over time, but any questions? And here's my email. And please contact me if you want to get involved. I hope you do want to get involved. Then just tell me, and then we make things happen. Thank you.